are stuck here.
And it was raining.
And that's basically the apocalypse. Like the freeway shut down. I don't get it. It's dangerous.
Like there was a crash on I-215 just today. So that's why it's that freeway shut.
So like anytime I'm driving and it rains, like the traffic just stops like very fast. It's scary. Because you're just driving and then all of a sudden just a wall of cars. It's
I got you.
You guys should at least be moving like 30 miles or 20 miles per hour.
Yeah.
Not just stop.
Well, so you know how in Michigan, on the road, the lines are reflective even when they're wet.
Yeah.
Not so here.
Yeah, that's what I'm starting to hypothesize. Is just their roads in Utah are not built for water. Like they deteriorate faster when it's
they have
wet.
standing water problems. They're like, I was driving back from Logan last weekend, and I was like, I can't even see.
And it's just as the lines are really straight anyways to begin with.
No.
These are Michigan. You're not coming to game night, are you?
That's a little change.
That's what Erik thought.
Is this study also in this
No.
system that you I saw so many times on your webpage? This what is
And Kevin's not going to be there.
the nematode?
C. elegans.
CA.
The first part will be, and then the rest will be
Okay.
in mouse brain neurons.
Okay.
So should I hook this thing up, or
Yeah, real quick before you begin, if you could.
because I'm super loud? But unless this is important for the recording.
Oh yeah, this is
is
only
only for
recording. The
recording.
microphone's in the
Okay.
in the room. Yeah, you're not amplified. Your voice. Is
Okay, because from teaching undergrads for years.
You got this.
No, I was teaching that huge one with 250 students, right? So it's like, you gotta scream. Yeah.
I haven't got
Thank you.
I haven't got used to that. Sometimes when I started speaking, but my voice gets low.
Oh yeah. And then people are what? Yeah. So are you introducing me? Is that?
Yes.
Okay. All right.
So I just copied this from your website. So that's on your website, and I see the genetic analysis in the
Nematode C.
nematode
elegans.
C.
Yeah, and I've been
elegans.
doing that ever since I was a postdoc, right? So
Yeah.
that's just, so no big changes.
Okay.
So yeah,
I'll
nothing.
just follow. Is there anything you want me to say
No.
besides things on your website?
Yeah, no, that's fine.
Okay.
No, and it's I've been here since 1994, and that's it. And in the Department of Biology.
And I'll say you are a professor in all the departments.
Yeah. Although that's less important. I mean, I'm here. I'm in biology.
Which of the awards are important ones?
None.
None?
None. I mean, the only one that is useful for this is the Humboldt. See, this is an old thing. I got a Humboldt fellowship, which allowed me to study in Germany for a year.
Oh,
God, this
it
is
in here?
old. No, this is really old. That's fine. I just
The Was that recent?
Yeah, just the last few years I've been working in Germany back and forth. So that's
Okay.
important because the people who, I need to give full credit to the people there who helped me out so much. So, it's,
Okay, I'll mention that one.
Or when you do the example of like how do you enrich uranium?
mute. Is this thing actually on? Yeah. Hey, how's it going? Michael's lab's here. I think we can start. How's it going, man?
Pretty good.
Good.
Good to see you here.
Yeah, yeah. No, it's what a whirlwind, man. The last month, I've been traveling the whole time. Just got back. Now I'm buried under all sorts of nonsense. Yeah, I think I have one trip from here till Christmas.
So the search committee is pretty much in place.
Oh, good. All right, excellent. So things worked out. Yeah, yeah. No, and Dave Blair is great. He's really, and when he's serving on two committees, right? He's serving on the biology search committee, and he's serving on.
Denise thought that he's never going to accept it.
Yeah.
I talked to him.
And he said, okay. No, he's just. I mean,
And I think it's going to be perfect.
and his senior member, and he's also extremely calm and extremely detail oriented, right?
The only person I'm kicking myself we didn't get is Joel.
Is who?
Joel.
Oh, yeah.
That would have been really
And he just said no. Yeah.
He's organizing some kind of an ACS meeting for 1,000 people.
Oh, that you can't let go wrong. That's good.
But I think the committee is very thorough and calm.
Yeah,
It's going to be fantastic.
good, excellent. No, and they're all sincere people, and you don't have any pain in the asses, so that's good.
Even Jim from Matt is very calm.
Yeah, Jim's great. Jim's like, he's the one guy who comes over and talks to me occasionally, and yep, it'll be good. Oh my God, the dinners, man. We have 3 searches going on, right? So we have the biology, we have the cell center, and then we have the biophysics. Come February, I'm gonna be so full of dinners.
I'm not thinking anything except at the search committee.
Yeah. Oh, we should wait a minute. Thanks. Familiar face in the crowd always helps. Yeah.
Actually, what is the face on there?
What is that? That's the neurosciences program's logo.
Whose face is that?
My wife made this. It's Julia Cameron. Was a 19th century photographer, and so she photographed Bloomsbury Group and all of that. So it was one of hers, and then there's a brain on top of it, as you can see.
She didn't photograph that.
No, she didn't. No, that was something new. But we used to have a lot of olfactory people on campus, and so the image is so olfactory, right? This prominent nose, and so all the olfactory people loved it. They go, "Yeah, it's the greatest logo ever."
So your wife does this kind of thing?
Yeah, she's a graphic artist. Hi, how's it going?
I now have a weapon.
Yeah, I'm looking out for that. Doesn't shoot, does it? No, I've seen the scars on Michael's back, so I don't know.
Another guy, I had another one. He grabbed it and it turned out it was a 327. It was not a very good weapon.
All right. As long as you're...
This is better. This will work better.
Okay. Weighted at the tip.
So why are you wearing a callus? You're Jewish?
Yeah. No, I have to get it. It does, right? It does look, right? It's got that perfect. My part of my family emigrated to Israel, so yeah, but I'm not. I didn't convert. They did, but. What's that? Yeah, I deconverted. Yeah, but there's a balance in all things, right? So it takes a Dane to convert. When a Jew deconverts, it's just really bad.
So have you heard anything from Mike these days?
I've talked to him a few times on the phone, but it's been all science. Like, when are we going to get this paper published? Before I used to get the litany of, oh my God, how things have been such a disaster.
You were writing 5 years ago?
Yeah, but I'm taking myself out of it. We have a collaborator who's really difficult, very slow. So, no, he has essential data.
No. What you do is very simple. You say, look, we're going to do it. We send you so-and-so data. If you want to input into the result, you respond by so-and-so data.
Yeah.
We'll send it off without your
Talk to Michael. He's been doing this endlessly. No, but I said I'm off. I go just.
But it's not going to do him much good now because you have to have paper.
Yeah, no. But I wouldn't be on the paper, so it'd be useful. I will take my name off so that he is a senior author on it. So because he's doing some of the stuff at UW. So yeah.
Anyway, I made a special question for you.
Okay, good.
Hello, everyone. My pleasure to introduce today's speaker, Erik Jorgensen. He's a professor here on campus in biology. He's also a professor in bioengineering, human genetics, and also is investigator in the Howard Hughes Medical Institute. He's the director of the Brain Institute here. Eric got his bachelor's degree on animal resources in 1979 at Berkeley. Then he did a study of genetics at Berkeley and molecular biology at University of Heidelberg in West Germany in the beginning of 1980. Then he did his PhD in genetics at University of Washington at Seattle. After he got his degree in 1989, after that he became a postdoc fellow at MIT, working with Robert Horwitz. And until 1994, then that's when he became assistant professor here, and he stayed until now. As an outsider, I can see that his research has been focused on genetic analysis in the nematode C. elegans pretty much all the time. And he has won many awards in research and teaching. He wanted me to mention the most recent fellowship, the Humboldt Fellowship, so that he can do research also in Germany. He has been traveling a lot in the past few years. So today, let's hear his research using electron microscopy to capture ultrafast memory movement. Welcome.
Thanks. I just wanted to feature the fact that I did a lot of work at the Charité in Berlin and I spend a lot of time every year there. And this has been a very productive collaboration, and I think when I get to that point, you'll see why it's so important that I thank the people who were involved in that. So the take-home message that I'd like you to take from today's talk is we've been trying to improve electron microscopy to really push it so that it's capable of resolving things at very rapid time intervals. So that's time, and so we can get this down to 10 millisecond resolution. And then finally in space, we're trying to localize proteins, and we can get this down to about 20 nanometers. And this is important when you're looking at very fast changes in cells. And so we knew that we had to develop these techniques. And so what I'll be talking about much of today is about the development of these techniques. I'll be telling you about the scientific results of those, but I would really like to feature the techniques because I think I can get feedback from all of you and maybe great suggestions as well. The scientific take-home message is these techniques have allowed us to discover a new form of endocytosis, that is, the recovery of membrane off the surface of a cell. And this occurs on an ultrafast time scale at the synapse. And so I'll tell you all about that so that you understand all of the jargon that I'm talking about. And so this talk will have three sections. The first is a history of synaptic vesicle membrane recycling. Then I'll tell you about our efforts to try to get precise timing of these events. And that's, I'll tell you about the discovery of ultrafast endocytosis. And then finally, what we need to do to move into the future is to determine the mechanism of these ultrafast events in the cell. And for this, I'll tell you about how we're trying to localize proteins to the sites of ultrafast endocytosis using nanoscopic fluorescence, that is super resolution methods, and electron microscopy.
So, Eric, would you tell us all what you mean by endocytosis?
Yes, I will. That I understand that's a piece of jargon, but in short, it means the recycling of membrane. So membrane goes onto the surface of a cell, and if you kept doing that, adding membrane to the surface of the cell would become big and floppy. So you have to pull that membrane back out in order to maintain the shape of a cell and its correct membrane tension. So that's what endocytosis means, but you'll hear much more about that later. So just to introduce you to the brain, our brain works by small molecule chemicals, and this is why drugs are so effective in treating brain defects. For example, in addressing pain, or caffeine, right, will make you more alert. Smoking cigarettes, I'm not recommending you go out and smoke cigarettes, but that also makes you smarter because of the nicotine. And the reason for this is that all of these produce small molecules that act on your nervous system in the same way your neurotransmitters do. And so here is an example of some neurotransmitters. These are the most abundant ones. GABA is the most abundant inhibitory neurotransmitter in our brain, and our brain works by a yin and yang of excitation and inhibition. The most abundant excitatory neurotransmitter is glutamate. Now, it's interesting that all these three, glycine, GABA, and glutamate, are all amino acids. And so you may want to think about that in regard to how did the nervous system evolve, right? Why is it that we use amino acids as the mechanism by which cells communicate? And so I could go on about this forever, but I'll just leave it for your own brains to meditate on. So, and this is how a neuron or nerve cell in the brain works. There is a cell body here, a long process. This can be up to a meter long that goes and targets other cells. And so the stimulation of this synapse requires synaptic input. So that causes this cell body to depolarize, which sends down an action potential along these axons. And this is highly reliable. It acts like a wire that will then connect to the next cell, where another synapse will cause this next cell to become active or inactive. So that's the basis of how the nerve cell in a brain works. What I'd like to do is focus on where the action happens, and that's here at a synapse. And so here's a blow-up of a synapse. Once again, the cell body is far, far away. So that's the mothership. That's where all proteins are synthesized, membranes are synthesized, and so those have to be sent down these long axons to the synapse. In some cases, these synapses are a meter long, and it would take a week using fast transport to get those components to the synapse. And so if you're trying to jump out of the way of a car in the parking lot here, you can't wait for a week to resupply your synapse. And so all of the components of the synapse must be recycled locally. So the way a synapse works is that there are these vesicles. This is the vesicle is made of a membrane, just like the cellular membrane is. It's filled with these small neurotransmitter molecules, and these then dock onto the surface of the plasma membrane. And they are now poised to fuse with that membrane and release neurotransmitters. So what happens is when that action potential invades this synaptic ending, calcium flows in, that causes these two membranes to fuse and releases the neurotransmitter molecules into the synaptic cleft. Those neurotransmitter molecules then bind onto receptors. They bind that neurotransmitter, but these are also ion channels. And so as soon as that neurotransmitter binds, that ion channel opens, and then ions flow through, and then can cause excitation or inhibition of this downstream cell. And so that's, in short, how the nervous system works. I told you that this is occurring at high rates, and you need to recycle components. So that means that the membrane that has been lost to the cell surface has to be recovered to regenerate synaptic vesicles. And so this process is called endocytosis. We then have to dismantle the scaffold around this vesicle, and then it's ready to be refilled and then reused. And so that mechanism is thought to be a clathrin-mediated process. So what's clathrin? Clathrin is a structural protein that builds that scaffold, right? So it builds a geodesic dome around a piece of membrane and generates a vesicle. And so this has been the classical model for about 45 years. And what I would like to tell you today is that we think is not happening. We think that there is an ultrafast mechanism that does not require clathrin. Okay, so the experiments that demonstrate this are shown here. So here is an electron micrograph of a point of contact between a motor neuron, that is a neuron that stimulates a muscle, and here is the muscle. You can see the actin-myosin filaments that generate that contractile force. And so at a typical neuromuscular junction, it can fire 100 times a second, and every time it fires, it loses some synaptic vesicles. So if you have 300 synaptic vesicles at a synapse, you can see that under these conditions, you'd rapidly exhaust the synapse of all of its synaptic vesicles and all of its neurotransmitter, and you'd be paralyzed, right? You couldn't move anymore. So this process of recycling was discovered by two scientists, John Heuser and Tom Reese, working at the Marine Biological Laboratory in Woods Hole. And the first experiment they did was they just stimulated it at very high frequency. So they 10 stimuli per second for a minute, and then they plunged it into glutaraldehyde. Glutaraldehyde is a fixative that has active groups on either side, and it cross-links all of the protein and stops all movement. And it also gives structural integrity to that biological structure. So now what you can do is you can embed it in plastic and then section it in very thin sections and look at it in an electron microscope. And this is what they saw. Here is a vesicle that is invaginating, a piece of membrane that is going in, and it has this coat, this electron-dense coat around it. And here is another one. You can see, again, this beautiful electron-dense coat. And here's one here that is a vesicle that's free from the membrane, and it also has this electron-dense coat around it. Protein. What's that? The scale, this is about 30 nanometers across, and so the whole structure here is about 500 nanometers across. So that just showed you that these, after intense stimulation, that membrane went back in. And so they said, so vesicles are recycled. They're not being fed from the mothership anymore. Is that like stained to get those images? Yeah, so this is This is staining with osmium. So osmium is electron dense, and it has 4 double-bonded oxygens, which are highly reactive. And what they do is they attack peptide bonds and attack free amine groups. And so proteins then become cross-linked. In fact, what it really does is it just completely degrades the proteins. And so you have this film, this spider web of connections that hold the whole cell together. But what it really prefers to do is to bind the double bonds, to react against the double bonds in membrane. And so these membranes are so black because they're highly osmicated. So osmium is, of course, electron dense. The rest of the proteins are sort of been completely eliminated because, as I said, it tears the proteins apart. So it sort of destroys the internal structure of the cell, but leaves really good membrane staining. So that's how that's visualized. Yeah?
From this micrograph, how do you know they're not docked? Like, what tips you off that they're coming back in and not docking?
Yeah, so in some cases they have stopped the stimulation and then plunged it into ice-cold glutaraldehyde. So you know that there's no more action potentials.
So what's the function of the glutaraldehyde, since you said that the osmium is really the thing that's doing it?
Yeah, so the glutaraldehyde goes in really quickly, and it has sort of a long chain, and so it can react against proteins that are somewhat far apart, and so they sort of cross-links.
The near-term stuff and the glutaraldehyde.
Yeah, the osmium is a fixative, but what we're really using it for is a stain. So that gives us something electron dense. Glutaraldehyde is not electron dense, but osmium is electron dense. I mean, since you guys keep stopping me and asking me really cool questions, I'll satisfy your curiosity. So it ends up that lipids they're unsaturated. So there's all of these double bonds in lipids. And so that makes them a little more fluid. And so osmium loves to grab those bonds and then react against both of them. And so then you have these lipids that are cross-linked. But then what the osmium does is it climbs like a spider or up the ladder, and then it ends up on either side of the lipid bilayer. I'll show you images where you can actually see that lipid bilayer stained with osmium. There's 20 carbons long, right? And you can see both sides of it. So it's exquisitely, it has very high resolution, right? So the detail in it is really wonderful. This you guys are really going to dig. This is the coolest experiment ever, in biology anyway. So this is these two guys. So Heuser and Reese said, all we see is we stimulate really hard. We see all these vesicles fusing. Let's see if we can synchronize it and determine the speed with which it happens. So what they did is they took a piece of a pectoral muscle from a frog, which is really thin and freezes rapidly. They left the nerve intact, and then they wrapped it around stimulating wires that then went up this long rod. And that rod was held in place by a solenoid, right? So it's just a friction hold. And then below this, they have a copper block cooled to 4 Kelvin in liquid helium. And so there's a shutter here, which they open. And then they release that starter solenoid, and then this begins to drop onto the copper block. Using standard physical equations of acceleration, they can determine 5 milliseconds before it hits that copper block, and at that point, there's a little trip that fires, and then it causes an electrical stimulation to go down the wire, which goes into the nerves, which then causes calcium to flow into the terminus, and it causes those vesicles to fuse, right? It required engineering, it required some understanding of physics, a great understanding of biology, and they managed to capture an event that people claimed never happened. And that is that these vesicles fused with the plasma membrane. And this was a controversy that extended from 1952 to 1979 when this image was published. And so here at 0 milliseconds is an unstimulated synapse. You can see that lipid bilayer I was talking about, right? That is 20 carbons wide. So you have this great resolution. You can even see it on some of these synaptic vesicles, right? And then if you stimulate 5 milliseconds before it's frozen, now you see these vesicles have fused. And here's one that's flattened out. The membrane's collapsing into the plasma membrane, into the cell membrane, and just losing all of those components, right? The proteins and the lipids that are in those synaptic vesicles. So this proved that vesicles fuse, and that was the big controversy, and they became quite famous for it. But what was more important is this, at least in my mind, the demonstration that there was recycling. And so here, let me explain what this is. You freeze a block that has a synapses in it, and right after stimulation or before, and you freeze it, and then you hit it with a knife from the side, and that causes the ice block to break. Where does it break? It breaks at the weak point, and the weak point is in that lipid bilayer. So these are just long carbon chains. There's no covalent bond that extends across that membrane, and so when you hit it, it breaks at the weak point, which is the place where there's no bonds, and so it breaks where the lipid bilayer is. Then they sputter coat it with platinum, and then they put that in an electron microscope. So you're looking into a synapse. Those synaptic vesicles are on the other side of the board here, and each one of those little buttons is a calcium channel. That's the calcium comes in and allows these membranes to fuse. At 5 milliseconds after stimulation, you see these little indentations appear, and on the other side is a vesicle that's just fusing. And because you can't follow that break into that concave surface, it breaks at the neck, so you just see these little flat surfaces. Then nothing happens. And then finally, after 30 seconds, you begin to see these indentations that are taking place here. And these, they think, are those clathrin, those coated vesicles that are being invaginated, where membrane is being pulled back into the cell.
So these are 3 different experiments of dropping this guy down.
Yeah, they did 100 of them, right? So they'd stimulate 5 milliseconds before it hit. They would stimulate it 10 milliseconds before it hit. What's that?
Have to load it all up again.
Yeah, it took years. And so then they would, finally they would leave it there for 10 seconds, and then they'd let it drop, and then leave it there for 20 seconds and let it drop, and then finally leave it there till 30 seconds, let it drop and freeze, and that's when they saw this event. And so they said, from this, endocytosis, membrane recycling, takes 30 seconds. It's a very slow process. It's not a fast process. Yeah?
Are you going to tell us what about the action potential actually initiates the membrane fusion?
Yeah, so I will not go into that. The action potential is just a way of sending a signal some far, far away, so you don't lose your signal over time, right? So the action potential regenerates an electrical response for a whole meter long.
Yeah, but what is it about that trigger which actually causes the fusion?
Yeah, so then there are voltage-gated calcium channels in the synapse. So that action potential hits that, and those things suddenly open. Because the gate is voltage sensitive, so there's part of the protein's charged, and it goes like that, and that opens the gate. Now calcium flows in, and then that causes the vesicles to fuse.
Yeah, because there's a change in pH or something, so that.
No, these are voltage sensitive channels.
No, I know, but I mean then.
How does the calcium do it?
Yeah.
So the calcium then has a particular protein that it binds, and that protein is a fusogen. So calcium is acting, so you have a negatively charged membrane, you have a negatively charged protein. Calcium binds and forms the meat between those two pieces of bread. And then that pulls that in, and that protein actually has these fingers on it that are hydrophobic, that want to bury themselves into a membrane. So when they go like that, they jam themselves into the membrane, and that pulls the vesicle, and that's the force that drives the vesicle into the membrane. This is still controversial. The version I gave you is one view of how that works. But Jim Rothman got the Nobel Prize a couple of years ago for a different model. Okay, so now we have a dogma. The dogma is this, that there's this protein called clathrin, that's the coat protein, and that accumulates around a piece of membrane, and that then pulls the membrane in, builds this Buckminster Fuller structure, the geodesic dome, on top of that piece of membrane until finally buds a vesicle. And then you have to pull off that clathrin scaffold to regenerate a vesicle that can be refilled. So that's how people have been thinking about this for the last 40 years. Process is slow, requires clathrin, takes 30 seconds. So now I'll tell you about our experiments. And so we wanted to revisit the Heuser and Reese experiments using more modern techniques. And the organism that we're using is the nematode C. elegans. And for these experiments, the important thing is it's normally touted as a genetically tractable organism. But for the experiments today, the important thing is it's transparent. This is an electron scanning EM, so it's covered with metal, so it doesn't look transparent, but it is normally transparent. So how are we going to stimulate this? Well, we want to do it intact. We want to do it in a physiological prep with under normal circumstances. It's not dissected out of the organism. And so we stole a little trick from Chlamydomonas. So this is a blue-green algae. And in daylight, it will go to the surface. Then at nighttime, it drops down so that it can protect itself from predators. And so how does it do that? And so these are just people who are studying weird things out there in the weird world. And they discovered that this blue-green algae has a light-activated ion channel called channelrhodopsin. It's related to the protein that's in our eye. That's rhodopsin. And that is not a channel. And so we still don't quite understand this. But this thing is a light-activated ion channel. So that here's channelrhodopsin, and this is just a tag, a fluorescent tag we added to it, that when light is flashed onto it, it opens a channel and sodium flows in. So positive ions flow in, and that causes the neuron to discharge an action potential. Right? So normally the cell is negatively charged, that keeps it quiet, but if you get a positive current, then that will cause these action potentials to fire. So now we can use light to activate an action potential instead of an electrode. The amazing thing about this is that the genetic code is universal, right? You can go to nematode, and the genetic code for making proteins is the same in a nematode, or you can go to a blue-green algae. The genetic code that instructs how this protein, channelrhodopsin, is made, it's the same, right? So you can just take, when you're doing genetic engineering, you just go to whatever organism you want, and you go, oh, here's a weird thing that they have. I'm going to take it, and I'm going to put it in pigs. You know, whatever you want, right? I mean, so you can just swap any protein from any organism into any other organism. Swap any gene from any organism into another organism. The gene makes the protein. So what we do is we express this in these motor neurons that contact these muscles. And this is in C. elegans. And if we have a dissected prep, we cut it open and we can put on a recording patch pipette onto this muscle. So now we can record electrical currents in this muscle. We stimulate with light. That releases neurotransmitter onto the muscle. And we can now record those events. So each one of these downward deflections is an inward current, right? So we have positive ions flowing into the cell. And here in blue, we're flashing that light at 10 hertz, and we're stimulating that neuron at quite high frequency. And so you can see that it maintains and follows that high frequency stimulation quite accurately. So we know that this works. The next thing we needed to do is to stop that after we stimulate once, right? Stop it at very rapid time intervals. And for this, we're using a high-pressure freezer. So what this device does, it goes from 1 to 2,000 atmospheres in 15 milliseconds. It goes from ambient to minus 50 degrees in 10 milliseconds. And what this does is, normally the water molecules are vibrating around, and as you cool something slowly, then they reorder themselves, and then they form crystals, hexagonal crystals. What that does is it tears the cell apart. So, you know, these people you read about in the New York Times who are having their brain frozen so that, 50 years from now we'll be able to figure out how to reassemble that brain and thought. Don't do it, because your brain will be mush, right? All the cells will be broken. Everything will be anarchy. What's that? Yeah, but that's not possible to do with a whole. We can get 200 microns deep, but something bigger than that.
The flash freezer for the brain.
What's that?
John is inventing the flash freezer.
Yeah, okay, I'll be your first, volunteer. Put my head in that. Yeah, you can, or high sucrose, you can do that, something like that. The one that was just in the New York Times, they waited until she, you have to wait till they die. You can't. But as soon as she died, there were people there ready to freeze her head. So they cut off her head and stuck it in because you don't need the body, you just need the head, and so they froze it.
That'd be a pity if she finds out later she needs the body.
Yeah. When she comes to, she might be disappointed in her new life. But this is really, this is predatory, right? These people know it's not working, and they convince people to give money. And then after a while, that heirs, no longer have money to keep the head frozen, and it doesn't work out in any circumstance. Okay, so we had to modify this little device. Here's the bayonet that holds this specimen holder, and this specimen holder is going to go into the high pressure chamber. Liquid nitrogen, the pressure will come in, hit this pin, and there's a cup here that will go drive it against a black diamond anvil, and then liquid nitrogen flows around here and cools it rapidly. So what we did is we had to get a light pathway into this device, into the high pressure chamber. So what we did was we replaced this black diamond anvil with a sapphire anvil. We drilled out that mounting screw, and then we drilled out this whole bayonet, and then we put an LED right here, so the LED has direct access to the sample cup.
Why is sapphire better than diamond?
Cheaper. You can get black diamonds for pretty cheap as well, but the sapphire, we lose the sapphire about 1 in 10 shots, but it's pretty strong. Sapphire also has this interesting property that it becomes superconducting as you cool it. So it actually draws off the heat much faster. So in some ways, it's advantageous over black diamond. But, clear is the most important thing, right? We could have a real diamond in there, but each experiment would be more than my grant. So here's a movie. This is Shigeki. He's going to mount the specimen cup onto this bayonet. The wire comes out and goes to a computer, and we control the flashing speed. So he's mounting it on a sled. The sled will shoot down this rail into the high-pressure chamber. And if you watch right here, you'll be able to see the flashes. He's going to start the program. Watch for the flashes right there. Boom, 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 boom. And then it shoots into the chamber, and then 2,000 atmospheres. With the original instruments, we still have some of them. You have to wear mouse ears. These are insulated. They're not so bad. It's still at MBL this summer. It caused a woman to fall off of her chair because she was sitting too close. Okay, so what do we see? So we see a very fast form of membrane recycling. This here is the structure called the dense projection, and this is where we think the calcium channels are. And you can already see this invagination forming. Here, that invagination is matured into a very large vesicle, much larger than the synaptic vesicles. And then this will translocate between 100 and 300 milliseconds into the middle of the synaptic terminus, where it is resolved. So it's great. We published that paper. Endocytosis occurs on 30 millisecond time scale, so membrane is recycled much faster than previously thought. Rather than 30 seconds, it's 30 milliseconds. But the reviews were interesting because they said, isn't this great? This little cute organism has this cute, very fast way of recycling membrane. And so people didn't believe that this occurred outside of nematodes. So it's clear we needed to find some other organism to demonstrate that this was true. And so for this, we turned to the mouse. And so the take-home message here is that we're going to see the same thing, that endocytosis, or recycling of membrane, takes 50 milliseconds.
And no mice were harmed in this process.
Mice were harmed. No pets or no horses or anything else, but mice did die. Okay, so this was done in Christian Rosemann's lab. So he heard my talk, and Christian said, I have mice. I have a high-pressure freezer. Why don't you come to Berlin and do your experiments here? So for the last 3 years, I've been going back and forth in Berlin doing these experiments. And my secret weapon was Shigeki Watanabe, my graduate student. He performed morphometry on over 10,000 synapses. He made 361 tomograms, which are these tilt series for you use an electron microscope for, and then he did serial tomograms to complete 11 complete synapses. And when you do this morphometry, you have to get your drawing tablet and you have to draw every membrane, trace every synaptic vesicle, measure the distance to the plasma membrane, see if they're docked. I mean, it takes a long time. So Shigeki doesn't sleep, so it's not a problem. The cultures, what we're doing is we're taking a mouse hippocampal cell neurons. This is part of the brain. We dissociate them and we put them in a culture dish and we give them whatever they need. They're perfectly happy. And they form connections between each other, and these are active synapses. You can put an electrode on two cells and you can see them talking to one another. And again, we're using the same method to stimulate. We're using channelrhodopsin. It's a different variant of it. We put that in a lentivirus. We infect the neurons, and so now those neurons are expressing channelrhodopsin. And the great thing here is that we're only doing one stimulus instead of high-frequency stimulus like was being done by Heuser and Reese. We're not using any drugs to try to increase the amount of calcium coming in. And we're using physiological levels of calcium instead of 10 millimolar calcium. And here's what these experiments look like. So this is a neuron that's expressing the channelrhodopsin. And we'll stimulate it with light. And this is just an on-cell electrode, and we can see whether it fires an action potential or not. So we flash the light. Here is our 10 millisecond light stimulus. And here is one action potential. Occasionally we see a second action potential, like we see in this example. But most of the time we just see one action potential. Then what we can do is find a cell that's not expressing channelrhodopsin. We can put our patch pipette on that and break in. This is a whole cell recording. We flash, and now we can see these inward currents. These inward currents are caused by the opening of those ligand-gated ion channels, by those, in this case, glutamated, activated ion channels. So that only occurs at synapses. And so we now see this positive influx of sodium and calcium into the cell, and so that's what this represents here. And this one is, again, an example where there are two action potentials. And so this looks like a synaptic current. This looks like the neurons are behaving like they should. To prove that, we blocked the action potentials with a drug called TTX, and that eliminates these postsynaptic currents. We added drugs that block those receptors on that postsynaptic membrane, right? They're the ones listening for glutamate. And if we block those, we also block that postsynaptic current. So this really requires action potentials, really requires ligand-gated ion channels. So it really looks like normal synaptic transmission. So now the experiment is to freeze at different times after stimulation and see what happens at these synapses. And in each time point is 200 synapses with a control. They're shuffled, and we're doing all of the analysis blind. So the results are reliable. This is what a hippocampal neuron synapse looks like, right? So the hippocampus is a part of your brain required for learning and memory, to acquire new memories. And it's a place for temporary storage until you go to sleep at night and you dream, and then permanent memories are made during that period. So here is a reserve pool of vesicles. Right here is one of these docked vesicles. You can see that. And this area here, this point of contact, is called the active zone. The active zone is just that. This is where vesicles fuse. This is the active part of the synapse. And then on the other side is the structure that's quite prominent. It's called the postsynaptic density. And this is where all of those glutamate receptors are that are waiting for the release of glutamate from this side. So now I'm going to show you some electron micrographs. So I just wanted you to understand what you're looking at. So where do vesicles fuse? They fuse in the active zone, like I just said, right? So here is one of those vesicles that's docked. Here's one that's fused, and that's at 15 milliseconds to 30 milliseconds. Here's another one that's fused, and it's flattening out. We're so proud of this image. This is the first time this has been seen since 1979, so it's really cool. And so we know the vesicles fuse, and we know where they fuse. They fuse in this active zone. Where are the vesicles recycled? not in the active zone. And so this was important for some other alternative models for how recycling occurred. So here is the edge of the active zone. Here's the edge of the postsynaptic density. You can see that there's an invagination occurring here. Gets deeper. Here's one that's very deep, and in fact, you can see this electron-dense ring that's beginning to form around that neck, and that will sever that neck. And then here's one that's the vesicle severed from the surface, and it will then move into the middle of the synaptic terminal.
So, Erik, how do you know what's the direction of time?
Yeah.
Snapshots,
Yep.
right?
Yep.
You're
Yeah.
looking at it backwards.
Yeah, no, this is so electron microscopists love to do this. So it's not the same vesicle, right? This is a whole bunch of different samples that are frozen. And then what we as electron microscopists do, say, I could arrange these into a flip book. And so you get them all stacked up and you go, and it goes right in and you go, wow, this is really great. In the C. elegans experiments, we had enough temporal separation that there was very little overlap between the different stages. So that wasn't a worry. Here, it was a big problem because there was, in fact, enough overlap that we couldn't be absolutely sure. And so, the point here is that this vesicle approaches the membrane, it fuses, and then it flattens out, right, if you reorder those images. We have an experiment that I won't tell you about, but what we added was a fluid phase marker. So there's something around the cells that the cells don't have in normally. They're little ferritin electron-dense balls. And then you can see them go in, and then finally they're found inside this vesicle. The only way they could have gotten in there is by endocytosing and going in. So absolutely great suggestion. How fast is it? It's, again, quite fast. So 50 milliseconds, we see these invaginations. 100 milliseconds, a deep invagination. This one's being cleaved. And at 300 milliseconds, we see these vesicles beginning to translocate into the middle of the bouton.
So these are all different vesicles, they're not just the same one.
Yeah, so I mean, when you freeze the sample, no more movement. And then we embed it in plastic and we look. So it's only by looking at 200 samples at each time point that we say, okay, in this time point we see these shallow invaginations. Later time point we see these deeper invaginations. But as I was saying to Carlton, it's not
on the fly without freezing. I mean, that is for the future, I guess.
To watch them in real time. Yeah, you need a super resolution microscope that is beyond the capability of existing ones. But Erik Betzig designed this new microscope called a Bessel beam lattice light sheet microscope. And this has millisecond time scale. It has structured illumination resolution, so 150 nanometers. Still not probably what we need, but I think we're getting close. So that would be the final goal, is to see this live. Okay, so what we can also do is cut thick sections and then tilt them in the electron microscope and then calculate the densities at each tilt series and then ask the computer to reconstruct where those densities are. And then once you have that slab reconstructed, then you can go through and cut virtual sections at 1 or 2 nanometers and then see what that synapse looks like in 3D. And that's a couple of these images here. The important thing here is at 50 milliseconds, we still see some of these vesicles fusing. But you already see these invaginations taking place at the lateral edge of the active zone. Here is another one of those shallow invaginations, and here's one that's really deep. It's about to be cleaved. Here is a synaptic vesicle that's docked to show you that these are much larger. So endocytosis it seems to be occurring at a much faster time scale than previously thought. Instead of taking 30 seconds, it's taking about 50 milliseconds in the mouse. So this is much different from what was seen before. What happens to these vesicles after they've been endocytosed? Do they regenerate synaptic vesicles? And the answer is yes. And I won't go through all the details, and this requires using the ferritin as a target to watch the membrane move in. So fluid phase marker. But this is one of these tomograms. This is a 1 nanometer virtual section. And you can see one of these large invaginations, these vesicles, are beginning to be torn apart. And so there's budding that's occurring. And there's actually a dense coat around these endosomes. An endosome is simply a large membrane-bound structure inside the synapse. It's thought to arise from endocytosis, hence endosome. Here's a reconstruction of one of those endosomes. And if you look at the electron densities in the top 20 nanometers of this, you see this hexagonal array. And this looks a lot to us like clathrin. And so we've done a number of experiments by knocking out clathrin, and we are quite confident now that this is clathrin. And so our point is that clathrin doesn't work during endocytosis. It doesn't recreate synaptic vesicles from the surface, but actually is recreating synaptic vesicles from these synaptic endosomes on a very fast time scale. So just to summarize where we are with...
Sorry, that second step, does that have to be fast?
No, but it is. Yeah.
Pulling apart the endosome.
Yeah, so what we see is that this process, 50 milliseconds to make a vesicle, after 1 second, these vesicles fuse to the synaptic endosome, make a large structure. Then they are budded by clathrin, and that takes 3 seconds, which is actually slow, but much faster than what had been previously seen. And then it takes about 5 seconds to remake synaptic vesicles. So it is a fairly fast process. Yeah?
How do you reconcile this with the Heuser
Yeah.
30 seconds?
So that's a whole nother talk, and then I left that out. But we think we know why. So when this paper was published, the reviewer said there is 40 years of work and over 10,000 papers that say that this paper is wrong. And there's one paper that says that this is the way things work. And it was a good point. They said we needed to establish why everybody else sees it gets a different result. The answer is temperature. So everybody does their experiments at room temperature. And when we do our experiments at room temperature, everything collapses back to clathrin-mediated endocytosis, clathrin-mediated recycling of membrane, and it's a slow process. So we can replicate their results. But mice live at 37 degrees, and so do we. And so if you do your experiments at 37 degrees, then you see this ultrafast mechanism. So we think we understand why. And now there's still a lot of skepticism in the community, so now we're walking through a bunch of different organisms, different systems, to show that, in fact, it is true more broadly than just in nematodes and mice.
So what is it that mediates the pinching off if it's not clathrin?
Yeah, so that's a perfect segue to all of the things we don't know. So what we now have to do is also show what the mechanism is. Because this is a novel mechanism, and I think that until we do that, skepticism is warranted. So what we have to do is figure out the proteins that are doing this. And so this is what this last part of the talk is about, is how do you figure out where the proteins are and which ones are involved in this process. Right, so the issue is where are the proteins and which are the ones that are associated with these endocytic events. There's this dense projection which marks where the calcium channels are, and so this defines really where vesicles fuse. And the protein that's in here, one of the most abundant ones, is called liprin. So our starting point was, can we identify where liprin is? So, and again, we can take jellyfish genes and put them in a nematode or put them in a pig, or eventually people do put them in humans. It's tattoos. You can get a green fluorescent protein tattoo. So we will tag liprin, and we'll attach it to the green fluorescent protein and ask where is it localized to. So here's a nematode, and so we'd like to know where liprin is localized. And so what we're going to do is just look at this dorsal nerve cord. This is where all the synapses are to muscles on the dorsal surface. And so here is the result of that experiment. We see beautiful dots where liprin is. So is it at the dense projection? I don't know. Right? We have no context. We don't know where the dense projection is to say whether this is at the dense projection or not. And so what we wanted to turn to again was electron microscopy, because it has extraordinary cellular context. So this next image I'll show you is a cross-section through the worm, and we're just going to be looking at the ventral surface. They have something like a spinal cord, except it's on the ventral side. So we embed the worm in plastic. We cut thin sections. We cut 30 nanometer sections. If you do the whole worm, that's some 12 to 15,000 sections. We only look at a few of them. But this is biology, man. Check it out. This is a high pressure frozen sample. We've never seen anything like this before. Here is this beautiful neuron cell body. It's round. All of these axonal processes, those are the wires that are going to the next cell. Those are all round. Normally everything's shrunken and collapsed on itself and you can't tell anything. And here is a synapse that's forming onto a muscle. So this motor neuron is going to stimulate that muscle and cause it to contract. So these are the structures we care about. So now we have 1 nanometer resolution. There are methods to look for proteins in electron micrographs, and that's called immuno-EM. So you can take antibodies that have a gold particle on them, add to the surface of the section, and see whether you can see where those proteins are. It's a horrible method. The morphology is, this is a tear. The membranes are not preserved very well. You have to do that, otherwise the proteins are completely blasted. And then you see a couple of dots and you go, yeah, that protein should be there. But then you see dots here and dots there and dots everywhere. So it's not a great method. So one of the problems is most antibodies do not work on these sections, do not identify proteins with little bits sticking out. Second, you have to add a gold conjugated antibody, and so that gives you this displacement, so it's not very accurate. Most of your proteins are deep in the plastic section, so you can never get antibodies against them. And then, as I pointed out before, you have lots of antibodies just scattered all over the surface that are sticking non-specifically. But I told you we can put the jellyfish protein, the green fluorescent protein, onto any protein in C. elegans. So we can change its genome so it makes these fusions where it has the protein of interest attached to the green fluorescent protein. So why use antibodies, right? All we need to do is shine a blue light at our plastic sections, and we should be able to see where the protein is. The problem is we need to preserve the green fluorescent protein in plastic. And that is difficult because for electron microscopy, you need these anhydrous plastics, but fluorescent proteins require a hydrated environment. They like a salty water environment to keep them folded properly. We figured that out by using these old school 1960s plastics called acrylics, and we can add water to them. The other problem is osmium just tears proteins apart, and so the fixation destroys the fluorescent protein. But we're using mild fixatives and we're reducing the osmium. So we managed to get this to work. So now we can take our protein, liprin, right? Remember liprin? We thought it's at the dense projection. Here's our fluorescent protein, and now we can look at it in our sections. Now it looks like a transmission electron micrograph, right? But it's not. It's a scanning electron micrograph. This is the method when you want to look at an ant head, these beautiful pictures. But what we can do is we can take our electron beam and pass it over our sections and then collect backscatter electrons. So the electrons, if it hits uranyl acetate, will come flying back, and we can collect those backscatter electrons, which depend on the dense staining. So we scan across the surface. We get this sort of low resolution image, but it's still good enough. You can see there's the dense projection. So now is liprin at the dense projection. Here's the fluorescence image. It's lousy, and there's a couple of problems, but the biggest one is that we're now diffraction limited, right? We're using photons, not electrons. So this needs no introduction to this audience, but I'll just go through it anyway. So here's the green fluorescent protein, 2.5 nanometers in diameter. So that's the resolution we'd like to have, right? We'd like to see proteins in these sections. here's the scale, the green fluorescent protein, and then when you focus that on your camera face, you have a 444 nanometer spot from minima to minima. So this is not useful because when we focused our photons from our image, you just get this big, huge green blob over, and the more samples, the more photons, or the more fluors you have on the section, the uglier your big blob is. So we had to beat the diffraction limit. And in the remaining 2 seconds, I'll go through at least one of these methods to tell you how we managed to beat this. So the first one, there are 3 ways to break the diffraction limit. Stimulated emission depletion microscopy, photoactivated localization microscopy called PALM, or ground state depletion. And so since we're in Utah, I'd like to give things as religious analogies. Stimulated emission depletion microscopy reminds me of my mom, because she would always say, many are called, but only few are chosen. So how does that fit into STED microscopy? So you have a beam that is an excitation beam that is scanning across your sample. So if there are any fluors underneath that beam, they become excited. But in the nanoseconds required, before they release their photons, you come back in with a depletion beam that is a donut shape. And anything in that donut is then driven down to the ground state. So they're dark, and now you just wait until those last few release their photons. Since you know where the beam is, you can calculate exactly, or you can see or image those fluorescent molecules that are under it.
Your donut beam is really a cylinder, right? It's a cylindrical shape.
Well, yes, exactly.
It's not concentric spheres, obviously.
It is. There is a high intensity donut, but in the middle there's no intensity.
Yeah.
And so then what happens is, as you excite your molecules, they rattle down, and before they can jump down and release their photon, the stimulated emission drives that down to the ground state. You get your original photon out of it, and then the one that is driven down to the ground state.
My point is only that the stuff in the center is cylindrical in shape. Not
Yes, exactly. So if you have a thick sample, you're catching everything within that cylinder, right? They now have new ways to do this, to shape that so that you have a sphere. Yeah, so that you have only the small sphere in the middle that's...
It cross things and destroy the stuff outside of this, so that you can approximate a sphere.
Yes, it's an approximation of a sphere. It's been incredibly successful. So here's histones. Histones bind DNA. You can see this is this is not a STED image. This is a confocal image. But we're cutting our sections at 50 nanometers. So we've already beaten the diffraction limit in the Z dimension, just because they have to be confined to this 50 nanometer plastic section. So this is way better than you get with any confocal microscope. And then when we apply STED to it, you can see that there's these clusters. Here's our EM, or SEM, and then here's the superimposition. And you can see the histones are in the area of the nucleus where DNA is. The nucleolus does not have DNA. Here is a mitochondria. These are circular structures that provide the energy to the cell. Again, we're already super resolution in the Z dimension. Here's the STED dimension, so the STED image. Here's the EM, and then here's the overlap. So you can see once again that we can get very precise localization of proteins. Here is Liprin, and here is the STED image. Here is our EM micrograph. There's the dense projection, and boom, lands right on top of it. So I'm going to just summarize STED microscopy, then I'm going to jump ahead to the very end of the talk. So STED works great. It works with any fluorescent protein. It's bright, but it has many disadvantages. First of all, the practical resolution is 60 nanometers. It needs a high density of fluors in order to see to image. It needs high laser power. They're expensive, and we don't have one. So for that reason, we switched to PALM. I'm only going to give you the Bible lesson here. We call palm microscope. This is the Roman model, in which one molecule is called and then immediately executed. So here's we have a molecule that can switch between green to red. And so if we come in with enough, just a little bit of UV light, one of those guys suddenly goes, I'm it. I'm red. I'm a red fluorescent protein. So then you collect all of your photons from it until you bleach it out, and now it's gone. And so, but your computer knows where that is. So then you go back to the beginning, and then you call another one, and it jumps forward and says, yeah, I shall liberate you all. Then boom, and it gets killed as well. So then by the time you're done, the computer knows where all of the centroids were for your molecules, and it can generate a really nice image. So I'll skip through that. And there's some palm images. They work. But this is where we're at now. This is ground state depletion. So once again, just the religious analogy. This is the Hindu model. So everybody dies, but are reincarnated again and again. So now what we do is we take, this does not require a photoconvertible fluor. It doesn't have to change colors. Any fluor will work. And so what you do is you just blast it with blue light until they all go into a dark state. They go into the triplet state. But if you just wait long, we can hold them up there. We have tricks to keep them in the triplet state. And then you let one drift down. So one drifts down. We then collect all of the photons from it until it finally goes back into the triplet state. Then we go and collect another one. And we figure out where it is, and then it goes back into the triplet state. And then often that very first one comes back, and then we can collect more photons from it. And so the goal, or the advantage here, is we get many more photons for every localization. So that is much more accurate. Two-color imaging is possible, and we have less problems with drift and things like that. So we now use organic fluors instead of fluorescent proteins. This is even better. We get 500 photons in 20 milliseconds, so now it's more accurate. We use a 3D super resolution microscope. This is an invention of Jörg Beversdorf, and that gives us images where we can calculate in the sample, in the Z dimension, where our fluor is, so we can use thick sections. And just to demonstrate that we can do this, this is a synaptic vesicle protein, the kind of things we'd like to follow in this cycle. And this is cut through the nerve ring. This is the brain of the animal. And we can get really good localizations. And this is just to show you that it's a thick section. It's 250 nanometers thick. When it comes on edge, you can see that, yeah, it really is thick. And here is a TEM image. It's a thick section, so it's a little fuzzy. But we can drop those right onto synapses. We can now do this in two colors. And here is a synaptic vesicle cluster. Let me just blow that up. Synaptic vesicle cluster, and this is an active zone protein. So these synaptic vesicles eventually will fuse where the active zone is. And then here is the three-dimensional view of that. So we can now see where the synaptic vesicles are, and we can see where our active zone is. So now we can begin to put proteins into the synapse. And what we'd like to do, and we haven't done this yet, we're right at the brink. We can take these tomograms from the electron microscope, and we can see structures, like here's a vesicle fusing, here's a vesicle fusing, here's a vesicle fusing, here's some endocytic structures. And so the goal then is to take these 3D super resolution images that show where the proteins are and put them on to these 3D images of the synapse. So then we can stimulate at different times. We can see which proteins are recruited to which structures, and then ask what proteins are likely to be mediating these events. What is the mechanism of ultrafast endocytosis? And so with that, I'll stop, and I'd like to thank the people at the University of Utah who helped out. Photons, those come from Rob Hobson, Carl Ebeling, Manasa Gudetti. Electrons, all of the experiments I showed today were from Shigeki Watanabe, except for that 3D image from Eddie Huber. I'd like to thank my collaborators outside of Utah, Stefan Hell in Göttingen did the STED microscopy. Jörg Beversdorf from Yale built the biplane microscope in our basement. And then my hosts at the Charité in Berlin. And I thank you for listening. Saviz.
Erik, since I know you love theory, what's the theoretical limit of vesicle formation in 200 nanometers for any membrane?
Well, it depends on how much membrane. The question is, how many vesicles can you generate from a membrane of a particular surface area? And so it depends on how much membrane has been added. So the number of synaptic, it seems to be perfectly compensatory. So the number of synaptic vesicles, we've added how many vesicles are lost at a synapse, and then we look and see how many are recovered, how much membrane surface area is recovered, and it's compensatory. We lose 4 vesicles to the membrane with every stimulus, and we recover 4 vesicles on average. So that's it, but it's probably just however much membrane was added. So it's not picking up extra membrane.
You think it feels the membrane being added and then it somehow?
Great question. So what is the mechanism? So what you saw was that exocytosis, membrane is still being added to the surface of the cell while it's being taken up. So that means that this piece of membrane that is fusing cannot be the piece of membrane you're taking back out. So that means that there has to be some rapid communication between a vesicle fusing here and the site of endocytosis. So I imagine what that is slack membrane. So the way I imagine is you have a bunch of stevedores that are pulling on ropes, pulling cargo into their loading their boat, right? And they've got ropes, and we actually see those ropes. We think that that's actin filaments. So they're pulling until the weight is too much, until the force is too much, and then they're just stripping the rope through their hands. Then you add membrane, and then it's slack, so that they're reeling it up as fast as they can. That's kind of how we think about it. The other is a passive invagination. So things tend to be fixed to other things in the nervous system. So if you add membrane, it's got to go somewhere. And so this is the tectonic plate model, that there are fixed plates of membrane that are solid and cross-linked. And so that you have at the mid-Atlantic rift, you've got a weak spot and you have volcanoes. And that's what we have. We have the mid-Atlantic rift, and then membrane is passively being spun into it. So those are 2 models, not exclusive. I think both of them are happening. Michael.
Now that you brought up communication potentially also through the membrane, is there any chance that all of this is an artifact of channelrhodopsin being there?
Yeah, so this is the biggest question that we get. What we did is we've done this with electrical stimulation and recorded what our responses are. We stimulated with light, and the responses are the same. We alternate, and it's the same. We stack them on top of one another, and it's the same. There's no additional component added by the light. So we don't think so, but I really think that that's the comment I hear the most often. So we're now trying to build a light-activated electrical stimulus. So getting into that high-pressure chamber is this big around solid steel, and then it's got a little hole in the middle about that big. So what we're doing is we're building little circuit boards, and we have a capacitor on that. So when the light comes in, it will stimulate the capacitor, and we'll get a decaying field, which hopefully will cause those cells to fire an action potential. Then the light is not it's an electrical stimulus. So that, I think, is the way to deal with that. Saviz?
Pressure shock.
The pressure shock happens, but we're not looking till much later, right? So the light stimulation, it happens after the light stimulation. So the light stimulation, the events we all see are in synchrony with the light stimulation, right? We see different event, and it always correlates to the light stimulus. The pressure shock, we see vesicles that have already come in and are being separated apart when the pressure shock occurs. We do not see events associated with the pressure shock. So why don't I just, time has gone by. I just wanted to make an announcement. There will be a special seminar, an informal seminar in biology on Monday. This is Sarah Abramson. She was in on building the first structured illumination microscopes. She's the one who came up with this multifocus microscopy and she's visiting my lab from the Rockefeller, and I convinced her to give an informal seminar on Monday. That'll be at 12 noon in 210 ASB, so that's the departmental room like this in the Align Skaggs building. So I welcome you all to come. It'll be interesting if you like microscopy. She's a physicist. You might enjoy listening to her talk. So thanks.
Agreeing to give this talk.
Sure.
At that time we had the cancellation, and I know we had to get a look.
Yeah. So you had to jump on it. Thank you for the coffee. I will finish it.
Very interesting talk.
Thanks.
Slides.
Yeah, I don't know if it's people in this audience get much out of it, but
Oh yeah, that was awesome.
a lot of cell biology.
Where's the Judeo-Christian theology in your?
I gave this talk and I gave it at Tel Aviv. And I also went and gave the talk at whatever it is, that place up there at Golan Heights. This beautiful, and so they are all totally psyched, and they're going, "We want the Jewish model. We want the Jewish model." So then they're trying to come up, and I go, "You guys are inventing, biological mechanisms that don't exist, so that you can have the Jewish model in there."
It could be something with guilt.
No, whining is what they all said. Can we get it so that the vesicle whines?
You just have a model where something reconstructs itself after a few thousand years.
Yeah, that's it. The ultra slow endocytosis.
That's great. That was really good.
Okay, I forgot.
I'm seriously having a hard time with the mechanics of these very fast endocytosis. Like just because this has to be somehow fluctuation driven, no?
Yeah.
So those fluctuations on the membrane in that scale are not, how fast are they? I mean, we can, somebody should be able to crunch the numbers, no?
Yeah, it would be good to have somebody who's a theoretician who works with membranes, the argument is that the membrane has to be, the way I originally thought about this before we knew that they were remaking synaptic vesicles, is I thought what this was doing was simply making the membrane taut again, because the membrane needs to be as taut as a drum, because the vesicles are rounded, so they already are stressed and they want to fuse. But if you have a plasma membrane, that planar membrane that is just absolutely packed, then there won't be any hydrophobicity exposed. So the way I think about it is that you have to create some stress on that so that the vesicle will fuse. So I thought what was going on is that the membrane was just being sucked up and then thrown out later to try to make that membrane taut. But the idea would be that that membrane is slack, right? So suddenly you have this wavy piece of slack membrane that is no longer being subjected to the drum head. What that drum head is